just to remind you to print your lab report and bring it with you together to come to the practical class next week. Right? I don't see. Do you want to take a look of your attendance? 73, I already can memorize. 73173. That's your code. 73173. Alright, today, which is the part 2 for the bacterial growth. It's here. Right, the content of today's lecture that include you learn about the how to culture the microbe in the lab, including the batch culture. You will know what is batch culture and what is continuous culture, and of course the growth curve. I think you have learned that before, but today we just revisions on that. Finally, regarding to the culture of the bacterial, you need to actually know how to calculate the number of bacteria in the culture. All right, and the part 2, which is slightly different from this part. Part 2 is regard to the biofilm and then also quantum sensing. Both are related to each other. So there is a lot of learning outcome here which you need to know, meaning that this learning outcome is by the end of this lecture, you need to know. I do not know whether you'll be able to do it, but supposed to be, you'll be able to actually comparing what is the different type of the medium that used in the laboratory, and you also be able, there are 3 different, at least in this lecture, there are 3 different methods when you need to culture the bacterial on plate, meaning that on agar plate, and also describe the 4 phases of the microbial growth curve and what are they and what is condition for each of them, and finally to know the way of doing the direct cell count using the counting chamber as well as the microscope. And lastly, which is also very important, this one, you're able to describe what is the biofilm and also how quantum sensing, the mechanism and how quantum sensing involved in the biofilm formations. Okay. To start with, we're talking about, I think this is one slide, yeah, jumping. There are conditions for the bacterial growth, right? 4 conditions in order for the bacteria to grow, and the factor that affecting them, the growth of the bacterials. Of course, in the previous lecture, we talked about the environmental factor, if you remember, right, the chapter 4, which regard to the temperature and the pH, the water availability, the aerophile, remember, the salt, tolerance bacteria or micro, and oxygens, whether they are aerobic and anaerobic, etc., which has already been covered in chapter 4. So this is the environmental factor really determine the growth of the bacteria. Now today we have these 3, which is all regards to the whatever the nutrient that present in the medium or present in the environment, which actually support the growth of the bacteria. Macronutrient, micronutrient, growth factor. Macronutrient. Macro, does it mean, what does it mean? Does it mean big size molecule? And micro, which is small size molecule? Actually it's not. Macro here meaning that the bacteria require this type of nutrient in large amount. Okay? If it's micro, meaning that it's small amount that is being required for the growth of the bacteria. Sometimes it's also called as a trace elements. And of course, the growth factor, not all bacteria require growth factor, but some bacteria really like, they are a bit fussy. They require some growth factor to support their growth. The growth factor include vitamin, amino acid, peptide, purine, and pyrimidine. Those are the growth factor. That is something that supplement added to the medium to support the growth. Otherwise, they can't grow. So 1, 2, 3. So what are macronutrients? Macronutrients, as you can see, that are 1, 2, 3, 4, 5, 6, 6. They are macronutrients. If you see that carbon, oxygen, hydrogen, basically they are there, sort of making up the building block of any tissue, any living cells. They require carbon, oxygen, hydrogen, which is organic, making up the organic molecule. So there are the building block that is called building block here. I think they have different terminology when they try to mention these three. How many? 1, 2, 3, 4, 4 of them. And very basic building block for the bacterial cells, which is carbohydrate. Carbohydrate, you can find a lot of carbohydrate in there. Where is it? Cell wall, right? Carbohydrate also present in, let's say, the outer layer, the slime layer. The slime in the capsule, you find a lot of carbohydrate. Also, carbohydrate making up a lot of present in the cell cytoplasm as and part of the nutrient. No, and then what about lipid? Lipid you found, of course, in the membrane. It's a major, component in the membrane. So, carbohydrate require basic elements, carbon, oxygens, hydrogens. Lipid, making up the lipid, you require carbon, oxygen, hydrogens, as well as nitrogens, phosphorus, and sulfur, which include all 6 of them. Okay, those are the macronutrients. Right, nucleic acid. Nucleic acid, which is the chromosomal DNA. Chromosomal DNA is contain the carbon, oxygen, hydrogen, nitrogen, and phosphorus. So those are the elements that require to make the DNA. And finally, protein. Of course, the bacterial cells contain a lot of protein. The protein, those to make the amino acid will have the carbon, oxygen, hydrogen, nitrogen, phosphorus, and sulfur, all 6 of them. So those are the macronutrients which they are required to make very important building block for the bacterial cells. And not only that 6 of the elements, here we also have some other macronutrient or macro elements, which including potassium, calcium. Potassium is, or magnesium or potassium required for a lot of enzymatic activity, enzymatic reactions. And calcium, you remember calcium is present in the endospore, which are required a lot in the endospore. We found that calcium. Ferrous and ferrous, which is iron, is part of the cytochrome. Cytochrome present, it is used for the oxygen respiration, for transfer of the electron. All right, so that's cytochrome. And this for respirations, and also is in part of the cofactor for the enzyme activity. Now, going to the micro elements or trace elements, there are some few of them here. These are required mainly for the enzyme or be part as a cofactor. So they more or less, some of them they needed to actually to perform the enzymatic activity. And this one, interesting, this is growth factor. Growth factor, it is essential to certain bacteria, but it's not for some. So growth factor include all this. And one of the example is the Haemophilus influenza. And what happened is that it requires 2 growth factor. One is called hemin, or is also called X factor. The other one is called nicotine amide adenine dinucleotide, NAD, or is called V factor. So if you actually has a plate, actually is commercially available to identify the Haemophilus influenza. You can use it to identify these bacterials by having this. This is just the, what is that? The V factor only. This is X factor, X factor only, and this is a V and X factor both present, right? So only this, there is a growth of the bacterial Haemophilus influenza. Well, this is a blood. This is a blood. Blood contain both V and X factor, but it couldn't grow in it. And the reason is that the NAD, which is the V factor, is able to be released by the bacterial, by the red blood cells. Sorry, is a blood. Red blood cells into the agar, so the bacteria can actually uptake it. However, hemine, which is actually inside the red blood cells and is not being released, so if the cells is not being lysed, basically the bacteria not be able to obtain the hemine. So therefore, there can no growth on the blood agar. However, if you're using a different type of the agar, which is a chocolate agar, it called because it look like, the color look like chocolate. This chocolate agar basically is a red blood cells, but the red blood cell has been lysed so that everything has been released out of the cells and is including the hemine. So on the blood agar, the Haemophilus able to grow as compared to the blood agar. Another way that you can actually have a growth of the Haemophilus on the blood agar is that you will have a streak, right? A streak of another bacteria called Staphylococcus aureus. Staph aureus able to lyse the blood. So when they lyse the blood, of course, they release the hemine. So when they release the hemine, and therefore there is a lot of the small tiny colony, they are the Haemophilus influenza, be able to grow along the line of this Staphylococcus aureus. So there's a lot more here. Okay, that's the way that you can actually observe the growth of Haemophilus influenza. All right. Now, the culture medium. The culture medium, it can be used, of course, the first thing for the growth of the microbe. The second thing, you use the medium when you do the transportations. You need to keep them something. Of course, you can actually, to dry the bacterials on paper and then revive them later, but this is not a common way. If you do not, normally drying method is for long term, but if you want to transport from one premises to another premises, normally you just keep them in a container containing medium and with the culture, right? And also you have the way to have a solid or liquid preparations, both of them. Now, when you're actually choosing the best culture medium for the growth of the microbe, you need to consider chemical compositions, whether you have to use define or have to use the complex, or physical nature, whether you should actually inoculate the culture in a liquid form, semi-solid or solid, as well as when the function of the medium, whether the medium able to support the growth of the microbe or is just keeping it as a minimum, not to allow it to grow too fast. So you can have basal, very minimum nutrient, just to require for the growth for the most of the bacterial, and some enrich, which you want to enrich to get it grow faster, and try to some bacteria not growing well in the laboratory conditions, so they have something enriched medium to help them to grow better. Selective, that is a purpose, we will talk about that later. Same for the differential medium, we talk about that later. All right. There are different type of medium that they have certain definitions, whether how you're going to use them and what is your purpose. Now, first thing is that you need to know what is defined, what is complex. Defined, as you can see from the slide, define, you have this recipe, right? So there is each of the component that to make this defined medium, which is called glucose salt agar. So all of them, they have a formula, the chemical formula, which is very well. So based on that, they have exact knowing what is this chemical and how much that you're going to use. So this is defined. Everything is very clear. So you know exactly what is the chemicals. However, for complex, meaning that, which is opposite to define, you got no idea somehow, right? What is inside the pectone, protease pectone, and also biosalt. These three doesn't have the very clear what is the ingredient inside these three. Basically, pectone is the extract that you extract it out of the meat, and then you use it to make the agar. All right. Same for this. Biosalt is also, of course, you know biosalt is coming from the bioduct. That's the biosalt. And this tree doesn't know exactly the very defined ingredient, and therefore biosalt contain a lot of like the acid, biosalt acid, and also this contain a lot of protein, peptide, lipid. Yeah. So, but you do not know exactly what is inside. So this is called complex. The example is such as McConkey agar, right? All right. The second one that you know is that how many different type of medium in their physical form that you can use. It depends on your purpose, really. For example, if I want to grow in the large amount, I need to extract certain protein out of the bacteria. I need large amount, really, very large amount. For example, 1 liter. This is 1.2 liter, correct? 1.25, let's say 1.25 liter. I need 10 liter of that bacteria in order to get what I want out of the bacteria. So what is the best way of culturing such a large amount? Agar, solid, this is an agar deep tube, broad, or slang, agar slang, but it's a different purposes. Whether it should be the broad or agar that you use to culture large amount of bacteria. Which is better? Of course, broad will be better, because agar, if you're using it, agar normally is very small. It's only 6 centimeter in the diameter. Of course, you can use 112 centimeter, which is slightly that big. But to produce something equivalent to 10 liter, I think you need a lot of this plate. So for culturing large amount, broad will be better. However, if I want something which is viable, I need a viable cells. I cannot rely on that, because sometimes you over culture it. It will ending up a lot of cell die inside the culture that you are not aware, because you didn't have any indicator. You didn't put in any indicator in the cell date. Therefore, if you want to have a very fresh, vibrant cells, you can use the agar plate. If it doesn't require a big amount, that you ensure you every single colony coming out, they are having a very fresh, good bacterial cells. All right. This two, I think it is having like, this is actually is the agar, it pull, I think it's solid, it pull until here. And actually you can culture something which is not the oxygen lover. For example, anaerobic bacteria, you can actually put the bacteria a bit down here so that they can grow without the oxygen. Right? So those are, this one, the slang is that they having a angle. So this angle basically increase the surface of the bacterial culture or colony growth. And this is also good for keeping the storage, because this is in tube form. And this is a plate. This is easier. Okay, this one is semi-solid, comparing to normally we grow bacteria using the solid agar, but this is semi-solid. The purpose is for the motility. as we know that some bacteria have flagella, they can swim. So if you want to identify if this is the bacteria which is unknown, I want to know whether it has the flagella or not. So I have to test on this semi-solid agar. So if this is a control, right? So I will actually insert a very sharp needle, something with the bacterials on the needle, and then I just insert all the way. Now, so it's insert all the way. So if you have something like this, that the bacteria is red color, so it's look red, easy to see. It's red color, so it doesn't actually spray. It's not like this. This is all spraying all over the agar. This indicates that the bacteria only localize at the point where the insert, right? So has no motility. However, this bacteria able to move, right? Diffuse, meaning they can move in the agar. They are motile. As simple as that. Now, further, there are different medium which used for different purposes. Basal, enriched, selective, differential. Actually 4 types here, not many, 4 types. Basal is those that just for the growth, that's all. A lot of bacteria, they can grow at the basal medium, such as the one nutrient broad or nutrient agar. Have you seen nutrient agar before? Yes, right? Don't tell me that you have never seen it. You have used it in your first practical class. All right, so these are in like something like this color. And they support most of the bacterial growth, but there are still some of the bacteria not able to grow on it. Enrich is for those bacteria are not very happy with the basal nutrient agar, so they need to be enriched by additional. So it's the nutrient agar plus something additional such as the blood, serum, or egg. So you have the blood agar, I think you have seen it before, but you have not seen this Lowenstern Jensen medium, which is like this in green color. So this Lowenstern or Jensen medium, or called LJ medium, normally they also call, they actually has this ingredient. All right, they have this asparagines, monopotassium, potato flour, and then important is the one highlight here, malachite green, which is give it a green color. So malachite green is also act as an inhibitor, allowed to inhibit certain bacterial growth. And glycerol will be something you'll be turned into carbohydrate as one of the energy source. Okay, egg base, they added the egg. Just is enriched, because this is also considered enriched by adding extra egg into the medium. Now, the next one is called this one here, McCongie agar. McCongie agar is a selective medium. What is meant by selective? Selective is it allows certain bacteria to grow, but suppress certain bacteria not to grow. So it's just favorable for certain bacteria to grow. So that is selective. Selectively favored to certain bacteria to grow, but not others. Antibiotic is one of the way, but antibiotic you can add into all medium. But this McCongie agar, if you look at the ingredient, where is it here? The one that I highlight, it is selective because it contains bile salt and also crystal violet. So bile salt is a salt. If you have I think 0.75 gram is quite a lot. It actually, high salt can inhibit also to get the protein aggregation in certain bacteria. All right. So what kind of bacteria is actually is good for gram-negative, but it inhibits the growth of gram-positive bacteria. Now you think about why it actually selectively used to grow the gram-negative but not gram-positive. You think about the cell wall. So what is the difference between gram-negative and gram-positive? So gram-negative contain, oh, I heard liquid. Gram-negative contain, still cannot hear. Contain what? I'm going to cry. I've been talking many times. Although I'm talking about 4 times in tutorial, but you only attended 1 time the tutorial. It contains the outer and inner membrane, correct, right? So gram-negative contain outer inner membrane, and this gram-positive doesn't have the outer membrane. That is the difference. Now we have to see what make them different. And for example, crystal violet is crystal violet is positively charged. So it will be positively charged, and somehow it's also killing the cells. It's able in large amount, if possible, actually killing the cells by interacting with the DNA. DNA is negatively charged. So actually it can damage or interfering with the cell division or DNA replications if you all the way go into the cytoplasm, into the nucleoid. So both of them actually is not very favorable to the gram-positive. And is actual fact, people say that gram-positive doesn't have the outer membrane, so it might be easy. There is no barrier, easier for them to passing through the cell wall and go into the cytoplasm. But however, the gram-negative have the outer cell membrane. May have something to actually to stop into to enter the outer membrane. It doesn't mean that it cannot enter, it's just slowing it down. All right? So McConkey agar is selective. It has its selections. It has a selections to favor to gram-negative, but against gram-positive. Now, we also know that McConkey agar has one more purpose, which is differential. So now what is differential? Differentials is that it allows the bacteria, different type of bacteria to grow on the McConkey agar. However, this McConkey agar allows to differentiate these different type of bacteria on the same agar plate. Here, important component is lactose and neutral rate. Lactose is used as a carbon source for fermentations. They use it to generate glucose and then use it for energy consumptions. Now, we have, cannot see that. We have the one that has the lactose positive. Let's say that lactose, when you actually utilize breaking down the lactose, eventually you will generate acidic product. Those acidic product will turn the color of the bacteria, the colony, into red. So the reason is because there is a presence of neutral rate in the agar. So this neutral rate is an indicator. If the bacteria produce a lot of acidic product, so here is all become red. So is the E. coli has most of them. This one may have a little less, but this is also a lactose fermenter. However, the one that doesn't actually turn into red, this is not a lactose fermenter. All right, so lactose negative. So it's able to distinguish these 3 different types. Depends on the lactose fermentations. One more, blood agar. I think you should familiar with that. So blood agar contain 5% of sheep blood normally. And how we able to differentiate is a differentiate. Same time is also a selective too. Is it selective? I think it's differentiate, not selective, sorry. It's a differential medium. And it has an indicator. The indicator is coming from the blood cells. Now, It depends on how the bacteria, how good they can lyse the red blood cells. So depends on the pattern of the cell lysis. They can call it beta, alpha, and gamma. Beta look like that. You see that all along the bacterial growth, the colony growth, is clear of the red color. So it's become there is no more red blood cells there because the red blood cell has been lysed. So it's have a complete hemolysis, all right, of red blood cells. So the example is Streptococcus pyogenes. You have alpha. Alpha is a semi-hemolysis, meaning that it's an incomplete lysis of the red blood cells, and therefore you will see something like green background, a little bit green, so indicate it's not totally lysed completely. And you have also gamma. Gamma this type of bacteria cannot lyse the red blood cells, so there is no hemolysis here. So it's called gamma. So it depends on the level of hemolysis divided to beta, alpha, gamma. Okay. We have one more here. This one is called mannitol salt agar. Mannitol is sugar. Sugar, what type of sugar? How do you call that? The one you use for diet. The one that you just don't want to have a normal sugar, you just put those no tablet. Keto. What is that sugar? Huh? Somebody say it. Whatever, I can't hear. Sorry. My ear, I'm not getting old. My ear are not very good. I mean, I cannot hear when your voice very soft. So when you talk to me, you have to yell at me, okay? So mannitol is used for that type of sugar. So it provides the carbohydrate. And here, it is a selective and differential medium as well, this mannitol salt agar. Why? It has a very high salt concentrations. And this high salt actually against most of the gram-negative, but some of the gram-positive. So the gram-positive such as Streptococcus. However, they are okay, Staphylococcus aureus, as well as Staphylococcus epidermidis. They are able to grow in high salt because they are the salt tolerance bacteria. So they are selective. Those bacteria cannot tolerate salt. They will not be able to grow, and mannitol salt agar. But for those Staphylococcus, most of them are able to grow. However, they also differential medium. The reason is that they have included the indicator phenol rate. So what is phenol rate do? Phenol rate, when you have pH 8.4 and above, you will red color. But if you have below 6.8, you have yellow color. So meaning that this is acidic, this is alkaline, correct? So acidic, why? Because Staph aureus is able to ferment the mannitol and then produce the acidic product, and therefore, because of the acidic product, that is become yellow. And this Staph epidermidis and not producing the acidic product, and therefore is still red color. So by this, you can actually easily differentiate between Staph aureus and Staph epidermidis. So, all right. We finished with talking all about different type of medium. Not many. There are many more because some of the medium serving very specific purpose, and it's not as common as the one that mentioned here. And this is the different sessions talking about isolations of pure culture. As I mentioned in the practical one, we culture the bacterial, right? We do the strict plate. The purpose is to get a single colony, right? The purpose is to get a single colony because single colony coming from the single cells. So within this colony, because it's originated from single cells, they all are taking one colony out to culture, you can get a very pure culture. So why pure culture is important, that you've been growing the pure culture in the lab. Normally, when we bring the bacteria or we bring the microbe into the lab is because we want to study them. Let's say that we're collecting the sample from the river, and because the river could be polluted, we want to know what type of bacteria like contaminate the river. Therefore, we collect the river sample, and then we need to isolate, seeing which each of the bacteria or microbes that are present in this contaminated water. So you will perform certain method in the lab to isolate a pure culture. So this is talking about how important that isolating a pure culture. I think that I have mentioned. Finally here, it is mentioned straight plate, spread plate, and pour plate are the technique used to isolate pure culture. Which one have you learned? Aaron? Straight plate? You have learned spread plate as well. Okay, I think probably you all have learned all three of them. But if I give you an exam, that you go into the lab having an exam, practical exam, let's say whether you can perform three of them or not. How confident are you? I'm not making any mistake, no contaminations. And then you're getting a pure culture, something grow on the agar. So this is something you need to actually revisit how we're going to perform this three plating method, and you have to do it properly and no mistake can be made. You're going to have it in one of your practical class. So this is basically a very, coming from the sample from the nature, it will be look like that if you're plating out. So this is impossible for you to study if you want to focus on one of them, very difficult. Therefore, we need to actually separate them, get a very pure culture. How do you do that? Of course, the method has been developed decades ago, and which is a very good way. We are still using this kind of method. The first one is the straight plate. Straight plate, I don't need to mention more. I think you have done it. You need to having a loop, flame the loop, and then you collect bacteria. So this is a single colony. You probably take this one, and then you into a new plate, you do the straight plate method. So this is so-called primary inoculation, secondary inoculation, tertiary inoculation. Why you want to do that? Here also show you this is good, this is bad. When you're having this basically, no, you can say that, oh, I got the bacteria grow, but it doesn't serve the purpose. The purpose is to isolate a pure culture at the end. How to get a single colony? There are certain things. First of all, when you pick up the colony, don't pick up the entire. If the colony is very big, don't try to grab everything. As long as you touch on the colony, I think you already got something on the loop. And then you just strike it, all right? Now, if you think you're taking too much of the colony, you can, in between, after this primary strike, you can actually flame your loop so that nothing left on the loop. And then when you do your secondary, you just cross, you just touch the primary inoculations, and then you do the second, okay? You just draw the line, so but touching the primary. So it's actually, you actually spreading the bacteria from your primary inoculation to the secondary. Same for the tertiary. And then finally, and then you just roughly do that. And with this technique, I can assure you that you definitely get single colony for sure. And why you will have some people getting this is very likely that the person didn't flame the loop in between each inoculation. Therefore, you must remember to flame your loop in each inoculation in order to get a single colony. So the single colony important, we can isolate this one for further study. Spread plate. Spread plate, you will starting with dilution maybe. We will talk about dilution later on. But the method itself is that you have a culture, right? You add a bit of the culture. Normally it's 0.1 to 0. Normally the best for me is 0.2 milliliter, meaning that 200 microliter. But 0.1 to 0.3 or 0.5, which is fine. Because if you have too much, it cannot absorb into the agar, which is you got very wet on the surface. So you need something that able to absorb quickly. So you have a spreader like this. This is a glass spreader or whatever. Then you need to actually spray it evenly all over the surface of the agar. All right, and then you incubate, and the next day perhaps you can get single colony out of that. Now the spreading, if you have nothing, you just turning the plate like this when you do spreading. But if you have something to help, this is called what? This is turntable. This is something that you can turn. So you just holding the spreader on the surface, touching the surface, and then you turn it, and it will turn. This one is quite useful. This one even better. This is automated. You just one touch, and you can just turn it by itself, right? So, but when you are doing it in your lab, I think you're going to turn it with your hand manually. Okay, make sure that you spray it well. And at the end, you will got something like that. But what we want to achieve is not too much of colony. This one is, first of all, impossible to isolate a single colony because most of them probably they're overlapping to each other. So you need to do a further straight plate in order to get a single colony. But this one, I think they are well separated. They have a lot of individual colony, even able allow you to count the number of colony that present on the plate. Counting the colony present on the plate is also very important. Somehow we need to do the counting. We'll talk about that later. All right, this is spread plate. Pour plate. Pour plate, I don't really like it. I don't think in my time when I'm working with the E. coli, I don't do pour plate at all. I do pour plate when I'm dealing with the bacteriophage. But pour plate, I don't do. But you will also do it in your practical class. How are you doing the pour plate? Of course, you will have a zero dilutions. You're starting with something, zero dilution, don't care. Okay, zero dilution there. And each of them you will do a pour plate. Pour plate is to get a molten agar, which is a soft agar, meaning that is already No, actually sorry, it's a molten agar, but it's not semi agar. It's something, it can be solidified, but it's just been melted. It's melted, you place it in water bath or 37 degree. After you melt the agar, you keep it warm so that it's still in the liquid form. And what happens is that you have the bacterial sample, right, where you want to plate it on the agar plate, and then you add this bacterial culture into this liquid form of agar, and then you mix well, make sure you mix well, and then you pour on the empty petri dish. After you pour it, and you just swirling it to make it evenly spread. And important is that after you do this, you don't touch it anymore to allow it, okay, to sit on the bench to allow it to solidify. That's it. And then you incubate that, and next day you can actually have zero dilution, and each of them you can collect a liquid or collect like a small amount, 1 ml, and then do the pour plate. And then eventually you can, at the end, you can actually see the zero dilution will give you a colony number Of course, if you have less dilution, you got more colony. You have more dilution, you got less colony. But eventually, you want something that allow you to see very clear individual colony that present on the agar plate and allow you to count the number. Okay, the one that been selected is this one. Then you can further do others. So this is pore plate, and the pore plate depends on, this is a zero dilution factor. The dilution factor is written here. This is less diluted, which is totally that the agar is covered by the bacterial. So it's a bacterial lawn rather than you can see a single colony. So this is more dilution, so you can see something that a lot of colony, and this is even having more dilution, so you can see individual colony present. So this is how it comes with the zero dilutions. Okay. Now, can you match each of this 1, 2, 3, 4 to 1, 2, 3? Which method? What is this? First of all, which method you're going to use this? Okay. 1, 2, or 3? Who think is number 1? Number 2? Number 3? Alright, at least not bad. Most of you think is number 3. So this is spread plate method. So this is spreader. So spreader, my time, we use the glass. We have the rod of glass. We just burn using the Bunsen burner to twist it, whatever, to bend it into this triangle. So we make our spreader ourselves. And now we have the disposable. This is disposable spreader. You only use one time. And you do not need to flame it. You just throw it. Okay? And what about this one? What is this? First of all, this is this is a water bath. Water bath, normally, of course, you keep the water and then you can turn it into different temperature so that you can keep the whatever you want to put inside the water bath into different temperature. So what is this used for? Who think is number 1? 1, 2, 3, a few of them. Number 2? Nobody. Number 3? Alright, good. So this is for the pore plate. Why we need to water bath? I think sometimes depends on the lab technicians. I think from the last practical class, you keep all the tube, right? Because you need to use the liquid agar for the pore plate. So we keep them in the water bath to keep it warm so that it's not solidified easily. So we keep them, and then you take the tube out of the water bath and use it for pore plate method. And what about this one? This is a petri dish. Who think is 1? 1, 2, 3. Who think is 2? 1, 2. There's some of them. You actually raise 2 times your hand. You say 1 and then 2, you also raise. All of them. Really? Okay. Number 3? Very few, but okay. Be careful, it is tricky. Sometimes the lecture is very bad. They give you a very tricky question. You see that there's no agar inside the petri dish. Empty, it's empty plate. So you shouldn't have, if you have agar, you can perform the straight plate on the spread plate. But this is the empty petri dish. This is been used, you have the liquid agar which allow you to pour into this petri dish. So this is used for the pore plate. You'll be given like this, alright, to you do the pore plate. And this one, I think everyone knows using this one, right? I think most likely is the straight plate because this is a loop and the loop have different size. This is the one which is very sharp, doesn't have a loop. Is Normally you use the loop to pick up the culture, and depends on how much you pick up, you have different loop. This is also disposable, which is plastic. You only use one time and you throw. Also quite expensive. And this one very sharp is the one to for inoculation. Doesn't use for streaking the plate. This loop used for streaking the plate, but this one is just pick up the colony and then it just dip into the broth and for culture, that's all. All right, good. You want to have a break? We can have a break. The break about 5 minutes to 7.5 minutes.
I told you, I warned you. Are you awake now? Are you awake now?
You know what? We have Google Translate.
Sure, let's go.
So ready for the second part. Uh-oh. Ah.
It's just one. Así va.
Alright, everyone good? Shall we start? So we can quickly go home after this. Alright, then the second part, we start with a different topic, which is the growth curve. This one is easy. Everyone know what is growth curve. So the growth, bacterial growth through the cell division. So this is just showing you how they can actually perform the exponential phases. 4 phases, the first one is so-called lag phase. What is lag phase? Lag phase is that when you enter a bacterial stock or bacterial inoculated into a new medium, so it's the bacterial will actually, that new environment or new medium trigger the bacteria to undergoing cell division. So before that, before they really go into cell division, of course they need to prepare themselves. How are they going to prepare themselves? They prepare themselves by preparing a lot of enzyme, cofactor, ribosome, which is those are the protein that require for DNA replication, require for cell divisions, all this. And the other thing that very important that happened during the lag phase is that if the DNA has any damage before they doing the cell division, they will correct the mutation. They will repair the mutations. Otherwise, if you keep going, there's a lot of mutation, it will cause the bacteria die at the end. That's why they need to actually, they have a repair system to repair the DNA before they really undergoing the DNA replications. So this is happened during the lag phase. Now, you go for log phase. One more thing is that duration vary in the lag phase, meaning that how long that the bacteria or the bacterial cells ready to undergo cell division, it varied around this time. All right, sometimes take a little bit longer for the preparations, and some of them is taking just a few hours. Some of them taking 1 or 2 days. Now, after the lag phase, you go to log phase. It's the exponential growth of the bacteria in terms of number. So in terms of the number of the living cells. And here, the growth rate is become constant, and the cell division also becomes constant because there are plenty of the nutrient that provide for them to growth. And once you reach, right, to a certain number, because the batch culture has the limited space, all right, also limited nutrient. Therefore, to certain time, they start to stop growing. So this is called plateau So the plateau is meaning that they are not growing anymore, but there are no cells are dying as well. So, or you can explain in a way the number of cells die and the number of cells growth is the same, is undergoing the same rate. So the gum is a flat plateau. And normally at this stage, right, is already start to have the not enough nutrient, not enough oxygens, and cells start to compete with each other, and also they start to produce the secondary metabolite. The primary metabolite normally happen in the log phase when they all require for the self-growth. But the second metabolite, okay, adds something else, and a lot of them undergo fermentation because of the lack of oxygens, and also they are producing a toxin. Remember that I mentioned that the bacteria can produce the bacteriocin, which is a toxin that against other bacteria. So they can probably produce here because they're going to kill other bacteria in order to survive. Okay, so this is the time. And then you go, of course, the dead phase where they start dying. So the number dramatically reduce. And all the nutrient exhausted, you have a lot of toxic waste, and the oxygens, no more oxygens. So that the number of cell dead exceed the number of growth. So it's actually dropping here. It's same as a growth and a dropping here. So one thing that important during the dead phase is the presence of persister, which are the cells that characterized by slow metabolic rate, persister. This is very important for certain infectious disease. So what is persister? Persister is a cells that they are having a low metabolic rate, meaning that they not produce very fast, so therefore they have a very low level of protein, and they are small in size normally, and they have a very slow growth. So if you're looking at here, this is actually the dead phase, right? So all the way the number is going down along the time. So this is when the cells are dying. Of course, those are the non-persistent cells. Those are the normal cells, which is in blue color. They start reducing number. But persistent cells, they are okay in keeping the number. So eventually, you will have the persistent cells survive at the end. And they have cosmetically, they are important because they can associate with chronic infectious disease, tuberculosis. Sometimes you thought yourself recover, the doctor given you a lot of antibiotic, you got a bacterial infection, so you recover, right? But after a while, like 1 month later, it's come back. It come back is definitely the bacteria are not being cleared from your body. So there are still bacteria there, and they can actually survive when under the treatment of antibiotic, and then they come back. So those are the persistent cells. When they come with the favorite conditions, then they will start to grow in the number again. This is a different topic, talking about the oven system. This is more, I don't think that you will be doing a lot with that unless you are studying biotechnology, which is very important. The oven system being used in the industry, growing large amount of the bacterial. Basically, they are not interested in the bacterial. They're just interested in the product that produced by the bacterial. A lot of them, they has been genetic engineered, and you remember the soy sauce? Soy sauce actually produced by the bacteria. Yeah, I think it's a Japanese company, they do that. And they are using this kind of oven system, which is a chemostat. They call chemostat as well. And what happens is that you have this, cannot see. You have the fresh medium actually coming in, and then you're also providing the air, the oxygen is also coming here. That's why you have bubble. So then it's actually circulating, right? And wherever you have the flow, something that's flowing out, which is the collecting here, the culture, same time allow them to collect whatever being produced inside their tank. So important is that the flow rate of the incoming and outgoing culture or medium, they actually in the same rate. So therefore, the volume inside the tank will never drop, will be maintained in certain volume. So it's maintaining the environment that all the time the cells inside the tank are undergoing exponential growth. So very simple if you're comparing both batch culture, which is a closed system, and continuous culture, which is an open system. So for the batch culture, you see that when you initially you have a lot of nutrient and then the nutrient drop, right? So then the products, basically it's a secondary metabolite. They produce, increase at the end, but that's it. After that, no more. But continuous culture, you actually maintain the substrate that provided into the culture, and also they actually consistently maintain as the same the production rate to make it. So this is actually important. Normally people using this continuous culture to study the bacterial growth in nature, because in nature you wouldn't have these kind of conditions. Occasionally, yes, of course, but most bacteria can actually have this continuous culture environments. So they want to, if you want to study the bacteria that survive in nature, then they will have to using this kind of system. Of course, they're studying the interactions between the microbes and different type of microbes and in the same system. Rather than this, because this actually give you very short time, not allow you to have a full picture of how different type of microbe interact with each other. Of course, finally, they are very important to in the food industry. But the cost to actually to have this kind of facility is very expensive. However, the long-term cost has become cheaper than this type of culture system. All right. Now, this is talking about if you have, let's say that if you have a sample coming from a polluted river, you want to know the number of bacteria that polluted the river. So you want to know the number. There are ways that you actually be able to count the number. Either biomass, basically you filter out all this debris and then you get the bacterial cells. You can actually removing, not even removing the water, no need to dry. You can actually calculate the biomass, just the weight, right? Or you can, if you want to know the Weber cells, you want to get rid of the dead cells, you just want to know the Weber cells present in your sample. Also, there is a way to calculate the Weber cells. And for most of us, we are interested in the Weber cells. If there are a lot of dead cells, we are no longer interested, because dead cell won't actually have any influence to the environments or whatever in the sample. We want to get is to count the Weber cells. So this is very common that when you do a sampling from soil, ground, meat, or milk, quality control QC in the food industry, yeah, they also need to, during the QC section, they also do the plate out to calculate the number. There are, of course, there are certain number. You allowed to have a minimum number, but you cannot actually over the threshold, meaning that your food product has been contaminated. Now, this is the method. There are several methods. Direct cell count. There are two types, hemocytometer and also the membrane filtrations, which is the direct cell count. And Weber cell count, turbidimetric, looking at the turbidity of the cells culture. So you got 1, 2, 3. Let us go to this one. I think we go through all of them. Sorry, we have to go through all of them very quickly. Have you seen this before? Yes, right? In your cell biology, we have that. You have a problem to see the grid, right? It's very blur. I do not know. I still do not understand. But the technicians say some of them are not very good, so it's blur. You have a hard time to find the grid. Now, luckily we don't have to do this in this semester, but we need to talk about that. This is the counting chamber. There are different, I think different brand. This is a Neubauer. I think it's a Germany brand. This one is a Petrohauser. This is a different brand. Whatever, they look more or less the same. It's just they are tiny different if they're different brand, but they look more or less like this. There is 2 counting chamber. Sometimes you want to duplicate your experiment, therefore you have allowed you to have 2 counting chamber. So you have the cover slip. So you put the cover slip on the chamber, on top of the chamber, and then you add your bacterial culture. Of course, the bacterial culture can be diluted already. Now, so after you cover with the cover slip and you add the culture, then they will actually present inside the counting chamber. And using the microscope, they're able to see the bacterial cells, right? With the grid that is very useful later for counting. And what you can see here, this is from the lateral view, and you see this is the slide like this, and then you put the cover slip on top, then you will have this culture. So it actually create a volumes in this. So you will exactly, when they design this counting chamber, they will exactly, when you actually put the culture, it gives you what is the volume of this. It will actually tell you when you actually purchase all this counting chamber. All right. So having this, you just do the counting. Let's go for the next slide and how we count. And you remember your your cell biology, which one you select. We call it grid. We have 9 grid, right, on one chamber. 1, 2, 3, 4, 5, 6, 9. So which one you use, you remember, for the counting? You remember? Which one? Excellent. You 0, 1, 3, 7, 8. But today we are not going to use 1, 3, 7, 8. We use the one in grid 5 here. We even use the one that highlight in red. The reason is that when we do the cell biology, we count is a human cells, which is much bigger. So we are using those has a bigger, the scale to do. But if you compare the bacterial cells, it's much smaller. So we use the one at the center. Actually, we're counting 1, 2, 3, 4, 5. And therefore, the counting chamber can serve different purpose for different size, cell size. Alright, let's go one by one. Now, hopefully you can see. You have 9 grid, right? 1, 2, 3, 4, 5, 6, 7, 9. And each of the grid, it actually can, if you put a cover slip there, each of the grid can give you about the volumes. So each of them will actually has 20 nanoliter. And for this number 5, grid number 5, each of the 25 square. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. Each of them, 1, 2, they are 25. We call this square. This is 1 square. Each of that is got 1.6 nanoliter. As I say that, the manufacturer already know the volume when they're actually making this counting chamber. So when we put on a cover slip, this particular red square, each of them actually in grid 5, they have 1.6 nanoliter. And then what happened that each of the 16 sub-square in each of the 25 square in grid 5 is 0.05. Basically, it's the small square inside this red square is 0.05. Forget about that. I think you can forget about that. You just remember of this. Each of these, the volumes of the culture, when you're looking at this area, is 1.6 nanoliter. And the rest of that, 1375, normally they also use to count the bigger cell size. That is about this volumes. Okay, you have two chamber. Let's say you do the counting for two chamber in the grid 5. So for the total cells that you can get in grid 5 is 1, 1. You're actually counting here, 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 and here. 1, 2, 3, 4, 5, right? And you will have 1, 1, 0, and the other chamber give you 1, 0, 5. Now, how are you going to calculate? Take a look at this. This is a formula. And adding all the cell number together, and then you will have, you divide it by, each of them have 5. The number of square, because 1, 2, 3, 4, 5 of them all together is 110 for chamber 1. So you adding all together, you will have 10 of this red square, and then the total number for 2 chamber is this. So at the end, if you this one divided by this, you will get each of this red square how many cells, average number of the cells in each of the square, red square. Now, you want it to be into nanoliter. This is 1.6. Each of these is 1.6. So you want to have the number of the cells in terms of per nanoliter. So you divide it by 1.6 nanoliter. This is the volume that for this particular square. So you divided that. Okay, now you get per nanoliter. Then you will also do the dilution factor, because you also do a serial dilution factor, which are not mentioned here, before you add it into counting chamber. So you have, let's say you already diluted 100-fold, you need to multiply 100. And then this one is the conversion from per nanoliter into per milliliter. So it times 10 to the 6. At the end, 1.3 times 10 to the 6 per mil. Sorry, I think I push. I'm not only pushing it, I also enlarge. My goodness. Okay. Go. You got it? Any questions? I do not think that you follow exactly, especially if you are very tired. Doesn't matter. You go back, when the exam comes, you look at your lecture note, you try to figure that out. If you have any questions, you come to me. But this is how the way, of course you have different way of counting it, but normally it's the same. It depends how much you have to multiply your dilution factor. And at the end, the unit is how number of cells per milliliter, which is, this is the number referring in your original sample. How much of cells present in your original sample. Okay, there are a second method for direct cell count. This is the membrane filter. You call membrane filtrations or membrane filter. So it's very simple. The idea is that you have collected your sample, right? Your bacteria, the culture sample could be from anywhere. And you have a filter here. This membrane filter is to capture the cells. Of course, before that, you could already remove the debris or anything else. So the filtrate will come out. The cells, the bacterial cells or any microbe will be trapped on the membrane. So one thing is that you can, before you do that, you can stain the entire sample with some staining method. For example, here is the Dabi. Dabi normally is a fluorescent stain to stain the DNA, to stain the bacterial chromosome. So after staining, you can clearly see the bacterial cells with blue color. So this one you already stain, and then you do the filtrations. So you take the membrane, you look under the microscope, you can directly count the cells with the blue color. So it's like having the blue color like this, more or less like this. So this is a direct cell count from your sample. Or if you want to have a wider cells, because in this directly counting right under microscope, some of them may be already dying. They are no longer undergoing apoptosis whatsoever. So they are dying, so they are not quite accurate if you want to just get the number of Weber cells. So in order to know how many number of Weber cells in your sample, you basically do the same, but you are not staining your sample with W. You just filtrate, get the membrane out, and place the membrane on the agar plate. And then you incubate the entire agar plate. So next day or 2 days later, normally you incubate a bit longer because some bacteria require time to grow. So you will have the colony grow on top of the membrane. So then you can count the number of the cells that they are considered viable. Okay. How you define viable cells? So this is, can you see that? This is how you define, right? This is alive. So alive is actively metabolizing cells. Then you will start to actually less activity. So cell with reduced activity, metabolic activities, then is that as a degree. If the cells still have some metabolic activity, but the plasma membrane still intact, but the RNA content reducing, meaning that they are in the shortage of ribosome or anything. So then it's getting worse if the cell are dying through this sequential flow, and then the plasma membrane is still intact but no detectable metabolic activity, and then the extensive damage of the plasma membrane, and then is cellular DNA degraded. It's all the cell pulse, and then it's considered dead. All right, this is how you consider. Of course, when we talk about Weber cells, these are the Weber cells, or perhaps include this and this. They still actively grow. Why we take so much effort to get the Weber cells and not consider the dead cells? Of course, the Weber cells will be able to grow and divide into bigger number. That, of course, that will affect whatever that we want to observe in the sample. Let's say it's a pollutions, we are only interested in Weber cells that will increase in number in the sample, right? The dead cells we will not consider because they are no longer important in pollutions. Therefore, the Weber cell count are very important. In order to get the Weber cells, to confirm they are still alive, metabolically alive, you need to grow them on the agar plate. Only on agar plate, with the presence of colony, in the day they have the ability to grow into a colony, then we consider a Weber cells. If you grow them in the broad, of course there is a Weber cells because the broad become turbid, meaning there is increase in cells number. However, inside the broad, the culture, they may have a lot of dead cells as well that we do not know. So normally when you talk about Weber cells count or Weber cell growth, we are talking about those cells grow on the agar plate and as presenting as a colony, right? Now, this is another way which I think you're going to perform that as well later in your practical class. This is another way of Weber cell count. You really count the number. How are you going to count the number? First of all, you will giving you a sample, the sample with unknown number of bacteria. So what you want your purpose, objective is to count the number of bacteria inside this sample, original sample. Now, we definitely know that most of the time, there will be a lot of bacteria, even millions of bacterial cells present in a small sample. We need to actually do a serial dilutions. The purpose of do a serial dilution, eventually when you grow on the agar plate, right? If you all directly use, this is already 100-fold. You still see a lot of the bacteria, not be able to count it. Only when you have reached certain dilution, then you'll be having a single colony, you're able to count 1, 2, 3, 4, 5. Okay, this is the purpose. That's why you do a zero dilution. Otherwise, no way that you can count with this amount. And you, of course, 1 in, I think roughly you know how they actually, this is 1 in 10 dilution, this is 1 in 100 dilutions. And each of them, there's a dilution factor that has been calculated. And this is important too, because how much that you spray, the volume that you spray on the plate is also important, because this number is reflecting by how much you spray on the plate. This is 0.2 milliliter, which is 200 microliter. And this is the calculations. Now we need to pick one of this which allow us to count. So which one? I think I choose this one. I don't know why. I think this is too little, may not be accurate. So I take this one, still allow me to count. And then I count that this plate has 150 colony. So I use this number for the calculation. And this is the dilution is around here, right? This is 1 in 10 to the 5. So the dilution factor is how much? 100,000. All right, this is the dilution factor, 10 to the 5. And the spreading of the on the plate is 0.2 milliliter. Now, taking this number, the colony is 150 colony that present in 0.2 milliliter, right? The one that we spray on the agar plate. So if I want to change this one 0.2 into 1 milliliter, so we can convert and it becomes 750 colony present in 1 milliliter, so that we can say 750 per mil. Of course, this one has been undergoing zero dilution. So if you want to track back what is the number, we need to multiply with the dilution factor, which is 10 to the 5. So 750 times 10 to the 5, and you will get 7.5 times 10 to the 7 cells per mil. That is the number of cells in your original sample. As simple as that. This is how you're going to perform the calculations. And of course, if you don't want to logically think how you're going to get to this, you can memorize this formula and use this formula for the calculation. Finally, you will have the CFU. CFU stands for what? Colony forming unit. So is how many colony? Is 7.5 colony, of course. And we know that each colony is coming from a single cells. So actually this reflects the number of cells present in the original sample. Good? Okay. Right, this is not a direct cell count. This is direct cell count? This is trying to look at, it's not a direct cell count. You don't count the colony, you don't count the cells under microscope, but you count the turbidity of the culture. And you're going to use this as well. I think these are using the spectrophotometric or spectrophotometer, which is look like that. Is very the principle, make it into simple ways, and you have this prism and monochromatic, doesn't matter, and actually they concentrate the light right into that. So your sample are putting behind it. So this is your sample with the culture. Let's say first of all you need to put a control, a reference. The reference which is just the medium without any cells. So with this as a reference must be 0 because there's no cells. So it will actually, the light easily passing through because there is no cells inside the medium. So you actually make it as a reference, meaning that it's a 0. And then you add your sample to this spectrophotometer, and now you have the cell particle. So when the light not easily pass through, it will deflect everywhere, different angle. The detector here will be converted into the reading. So this reading will give you 0.2, 0.5. The maximum can go up to 2. Anything that more than 2 point something, and then they are not readable. So you have to actually keep it between 0 to 2. So with that, we know the turbidity of the cells in the culture. Some people already calculated with this kind of this reading can convert into number of cells. All right? Okay. This is the very last one, last part, and also quite important. It's nothing anymore, not practical anymore. This is about the theory about the biofilm. Biofilm, as anyone of you know about what is biofilm? Biofilm is just, if you imagine you have seen colony. Biofilm is look like colony, but it's much more complex, and therefore we cannot call biofilm as colony. Colony is very simple, but biofilm is something that also starting could be from colony and then slightly develop into a biofilm. Because in the biofilm, they found there are a lot of this kind of so-called, this is something you need to remember, extracellular polymeric substance, EPS. Okay? So you need to remember, is the biofilm contain of this? What is this so-called extracellular polymeric substance? It not only having the biofilm having the cells, plus with all of this, it has polysaccharide, secreted protein, lipid, and extracellular DNA. So you need including the DNA present, forming a very complex matrix inside the biofilm. So this is the scanning electron microscopy that you see each of that. This is like a small hill. They are the biofilm attaching on the surface. So they may starting from small colony and become more complex and more complex. And they can consider is a one bacterial community. Right? Inside this community, they have their way to govern the community. Very complex, and they look like this. Now, before we talk about biofilm, we need to know that there are two ways that the bacterials, right, they can be presented as a planktonic, which is they are individual single bacterial cells, and the other one is that they can aggregate together into a biofilm. And this is a comparison between this planktonic, they are free living, right? Perhaps if they have flagella, they can also swim along it, and they are motile, normally it's sessile. They are actually attached to the surface, and they are more sensitive to antibiotic, they are resistant to antibiotic. That is the main problem of the biofilm, because they're resistant to the antibiotic. Low frequency of gene transfer, high frequency of gene transfer. What do you mean by gene transfer? Conjugation, plasmid transfer. In this biofilm, they are very clever, the cells. In order to survive, if one of the cells, bacterial cells inside the biofilm, they have the plasmid against the antibiotic, probably they have a very high rate of gene transfer for those antibiotic resistant gene, right? We also know what is persistent cells, right? Those can survive like dormant cells. They have a low metabolic activity, but they can survive and against the antibiotic. They have very high level of persistent cells as compared to the planktonic cells, and they can do finally quantum sensing, communications. In the biofilm, the cell to cells, they can talk to each other. So if they are present as individual planktonic cells, normally they don't talk. How do you measure the talking between the cells? You heard about quantum sensing, right? Yes? No. I take it as no. Current sensing, I think is a topic of research, but it's already quite some time. It's back to like 50, 60 years ago, people already actually focus on the current sensing. Current sensing actually is, I think we talk about that later, is a communications using the molecule, communication between the bacteria. But let's just focus on the biofilm. Now, the biofilm, right, they can actually attach to any surface. That what worry us is that if they attach to the medical device, we have those the patient who go to undergoing the hemodialysis, they have a catheter attached to the body. The catheter basically is a metal. Actually, it can, if they got contaminated, bacteria can grow and form the biofilm. Your contact lens, yeah, actually is also a good place for forming a biofilm. So you wash your contact lens every day, or you use a disposable contact lens. And those are the bacteria are commonly sticking on the medical device, and the Staph aureus, Staph Staphylococcus epidermidis, and Pseudomonas aeruginosa. Those are the three, because they are present on our skin. That's why we always like, they also opportunistic bacteria. Normally they are actually present on the skin, they are harmless, but when they form the biofilm, then they become pathogenic. Now, this is a community, a biofilm community. What happened inside? First of all, you can look at 1, 2, 3, 4, 5. Also, this is 5, also I think discussed in the textbook. Now, what is that they have like this big biofilm, but the oxygen level is different. When you are going towards inside, there is less oxygen. Outside, of course, is with the oxygens a lot in the environment. So you have more oxygens on the surface area, but less towards the center. Same for the nutrient. You will has accessible to nutrient when the bacteria are on the surface, but when you go in, those bacteria have less accessible to nutrient. Second, they have changing, a lot of things happen inside the biofilm. They change the physiology, and they build up a more organized community, including they produce the influx pump. They're pumping a lot of the molecule they don't want. For example, the antibiotic, they don't want, they have the pumping system to pump them out, right? They also have the transportation, a channel, because inside we have less nutrient, less oxygen, less water. They can have the channel for transportations that build up to transport water into the center. And therefore, a lot of physiological changes happening in the biofilm. The third one, the outside they consider is more negatively charged, and therefore, if the antibiotic is also negatively charged, there is a repulsion, right? They repel each other. That's not to stop or slow down the antibiotic for penetrating into the biofilm. Number 4, persistent cells. Yeah, persistent cells forming a very cells that is present, and they are very strong against the antibiotic. But they have found persistent cells in the biofilm. And lastly, what is that? Genetic diversity, meaning that biofilm are not like colony. Normally colony is actually homogeneous, only one type of bacterium, but biofilm can have more than one type of bacterium, different type of bacterium. E. coli plus salmonella, they are living together. So it's like a community can have different type of bacteriums. All right, so these are the characteristics of the biofilm. And to hurry up, I got another 10 minutes. Those are the formations of the biofilm. Looking at that, there are 5 steps. All right, so 1, 2, 3, 4, 5 is how to develop the biofilm. If you look at this scanning electron microgram, you see that you see nothing very, very actually special, because you only see a single cells that building up single cells like this. getting more and more. But in actual fact, when you study it, there is a lot of changes. Now, at stage 1, normally they are coming from planktonic bacterial, they are plankton. So they can actually attach to the surface and then they can detach from the surface. So this is so-called reversible attachment. They can attach and detach easily. So when you come for stage 2, basically now the attachment is very, very strong. All right, here is very solid on their surface, and then they start to build up, growth and build up. You come to stage 3, they build and formation of a micro colony. Right. Number 4, they become more complex, they start to get mature, and they have the EPS. The EPS, extracellular polymeric substance. We build a lot of carbohydrate, lipid, protein that's secreted by cells and exchanging the genetic material through conjugations and building a small community. Now this biofilm is getting very mature. However, because biofilm also, if the surrounding the biofilm, they have very limited access to the nutrient or water. So at one point, there is a dispersal, meaning that some cells, they start to detach coming out from the biofilm. And then those cells become the planktonic bacteria again. When they come to an environment which has nutrient or somehow better environment, they can detach again. So the development of biofilm is actually going in a cycle like this. All right? Okay, let's go. Next one. Now, we talk about the biofilm. When the biofilm, they become mature, they have detected, they actually has the current sensing. So the current sensing, as I explained, is a communications between the bacteria. They talk to each other. So what is the current sensing? Is using a molecule or molecular mechanism by the bacteria to communicate, right? So that they can collectively adapt to a behavior. And there are 2 prerequisite, whatever things that they need to fulfill in order to get to current sensing. So what is that? They have to fulfill 2 things to have the current sensing. First thing is the cell density. You need to accumulate enough of the cells, populations, then you can start to have current sensing. Otherwise, no. If let's say it's the planktonic bacterial cells, and the distance between cell to cell are very far away, so unlikely they can have the current sensing. Second, they need to actually release the molecule called autoinducer. This is very important. This is how I say that they communicate each other with this autoinducer. So without autoinducer, without expressing and released autoinducer, they cannot communicate. All right. And talking about their autoinducer, right? There is gram-positive and gram-negative bacteria. Both gram-stained bacteria, they can do current sensing. But the molecule that used for current sensing, slightly different. What happened is that in gram-positive, what do we call them? Let's see. This is called autoinducer. I think in gram-positive, they still call autoinducer. Those are, let's say, is signal peptide, and they actually secrete. Then there is a receptor. Actually, they can influence the molecule they secrete out, right? The autoinducer they secrete out actually can come back to themselves. So there is a receptor, a sensor to receive this autoinducer. Somehow, the autoinducer, the ultimate purpose is to turn on certain gene, to instruct the cells. Now we actually come with a very high density of yourself with other, then you should do something. So they turn on certain gene, so the gene will express and will change the physiology of the cells. Now for gram-negative, they don't call as an autoinducer, instead they call AHL. is N-acyl homoserine lactone. This AHL is formed by the homoserine lactone ring with the fatty acid chain. Actually, two components. This is an example, Vibrio fischeri, which is bacterials that can emit the bioluminescence. It's a bacteria living in ocean. So this is a example for current sensing. So this Vibrio fischeri is a symbiont as well, that living inside the squid. The squid that living inside called Hawaiian bobtail squid. It's very small. I think it's about kind of small as 1 centimeter. Some of them as big, only like 4 centimeter. Not very big, very small. This bobtail squid, in the daytime, they are actually inside the sand, hiding themselves. At nighttime, they're coming out, of course, for hunting, right? The prey. Now, they have a so-called light organ present here. These are the light organ. And normally, these Vibrio fischeri, when they have seen, they somehow, how do they attract to each other? How this Vibrio fischeri can go into the light organ? I think not much is being known, but a lot of people still studying it. Maybe that the squid release something to attract the fischeri to go into that. But somehow, when they see each other, they will all the way like coming to here, so that they get more and more bacteria coming to here, and there is a cilia flushing them go inside this. This is called intake chamber. Intake chamber is this two organ, right? So you have aggregate of the bacteria and go into sitting here. This is basically housing the bacteria and same time a lot of nutrient. So the squid provide the nutrient to the bacteria. Then what bacteria do to the squid? So what they can do? They can emit the light. This is when the current sensing come to play a role. Now, in the Vibrio fischeri, they have an operon. Operon has all these gene, Lux gene, Lux E, B, A, D, C, I. So this Lux I can produce AHL. AHL, as I mentioned just now, which is the autoinducer, right? The molecule for current sensing. So it actually is express the AHL synthase, which can produce the AHL. So when they have accumulate enough inside here, inside the antechamber, they actually in a high density. So the current sensing only require when the cells accumulate into high density. So they start to has to release this AHL. So the AHL release, it will release inside here, and then two other bacteria also have the receptor. It itself also have receptor. So within this small space, there is a lot of AHL. So somehow the bacteria have a receptor to receive the AHL, and the AHL will combine with the so-called LuxR protein and bind on the promoter for LuxR. A LuxR is a receptor for AHL. So they will generate more LuxR receptor, and also LuxI will generate more. And also all of these genes will be expressed. And one of those is LuxB and A, which actually responsible for the production of luciferase. You know, the luciferase is important to convert whatever the substrate into the light, the luminescent light. And with that, is fully expressed, activated this operon, and you got light here. So the squid will look like this at night. Why did the squid need this bioluminescence? It's some kind of a camouflage that allowed them to escape from the predator. So they actually they normally have a shadow, right? The shadow can be recognized by a predator, and therefore they can capture by the predator. If they haven't emitting the light by themselves, they can cast the shadow and then will not be actually found, detected by the predator. So the predator may think that they are the shadow from the moon, because the light and the color and also is look very much like the color from the moon. The intensity actually they can adjust to exactly on that particular day, the intensity of the moon that actually shadow on the ocean. So this is how it do. So actually it's a symbiont, mutual benefit each other. Okay. This is just a slide describe the whatever the previous slide. This is the last one. We have no more. Okay, last one. Current, what is that? Current sensing has been found to participate in this a few of the functions. Those are actually is a survival strategy for the bacteria. They are involved in sporations. They upgrade a lot of gene to make their spore. They can do the bioluminescence, the example given just now. They are actually increase the virulence gene expression to against the host defense system. They can do the conjugations and competence. They can also uptake of exogenous DNA, like taking the DNA from other bacteria and increase the competency. And also they contribute a lot to the biofilm formations. Alright? Alright then. That's all. This is the very last slide. Thank you